Welcome students. Today we will discuss different types of DNA and RNA. The objectives of our today's lecture are deoxyribonucleic acid or DNA, different types of DNA, ribonucleic acid or RNA, and different types of RNA. Let's begin with DNA. There are two classes of nucleic acids, deoxyribonucleic acid or DNA and ribonucleic acid or RNA. DNA is the hereditary molecule in all cellular life forms as well as in many viruses. It has two main functions: to direct its own replication during cell division and to direct the transcription of complementary molecules of RNA. DNA is made up of a strand of nucleotides, and each nucleotide is made up of a sugar, a phosphate group, and a particular nitrogenous base. The bases found in DNA nucleotides are adenine, guanine, cytosine, and thymine. British scientists Rosalind Franklin and Maurice Wilkins undertook the methodical work to photograph and analyze X-ray diffraction images of DNA molecules. From the nature of these X-ray shadows, Franklin was able to identify two distinct but regularly repeating patterns in the structure, one pattern recurring at intervals of 0.34 nanometer and another at intervals of 3.4 nanometer. Double helix model soon became established as the definitive structure for DNA. Watson and Crick postulated a three-dimensional model. It consists of two helical DNA chains wound around the same axis to form a right-handed double helix. The DNA molecule wraps tightly around groups of histone molecules in a regular pattern to produce bead-like structural units called nucleosomes. This short segment of DNA is bound to a single molecule of a type of histone known as H1. The interaction among H1 molecules helps to draw the arrangement into a tight regular array. In turn, this array, which has a total thickness of about 30 nanometers, undergoes another level of packaging. It forms loops that attach to a supporting structure of non-histone proteins. As the cell prepares to reproduce, the protein structure folds back on itself to condense the chromatin even further, resulting in short, thick chromosomes. The important feature of double helical model of DNA structure are supported by much chemical and biological evidence. Now coming to types of DNA. DNA is not exactly a uniform structure. A number of biochemical and biophysical techniques have shown that double-stranded DNA can adopt a variety of conformations. First and most common form of DNA is bDNA. DNA duplex model proposed by Watson and Crick is right-handed spiral and is called bDNA or balanced DNA. It is the most common form of DNA, originally deduced from X-ray diffraction of sodium salt of DNA fibers at 92% humidity. bDNA consists of a backbone of polynucleotides made up of alternate sugar-phosphate groups with nitrogen bases projecting inwards. Adenine of one polynucleotide chain is held to thymine of opposite chain by two hydrogen bonds. Cytosine of one chain is similarly held to guanine of the other chain by three hydrogen bonds. The pitch of helix is 34 angstrom with roughly 10 base pairs in each turn. The average distance between two adjacent base pairs comes to be about 3.4 angstroms. Another type of DNA is aDNA. aDNA or alternate DNA is right-handed duplex. Here a single turn of helix has 11 base pairs at low humidity, that is 75%. Compared with bDNA, A-form helix is broader. It is 2.6 nanometer in diameter, somewhat untwisted, with 11 base pair per turn, and the bases are tilted and lie well off the helix axis. The major groove is thin and deep, while the minor groove is shallow. Third type of DNA is zDNA. zDNA has two strands, coiling in left-handed helices with pronounced zigzag pattern in the phosphodiester backbone. zDNA can form when the DNA is in an alternating purine pyrimidine sequence. zDNA is like a helix of dinucleotide pairs. zDNA essentially lacks a major groove and has deep and narrow minor groove. Fourth type of DNA is cDNA. This form of DNA is formed at 66% relative humidity in presence of lithium and magnesium ions. cDNA has 9.33 base pairs per turn of spiral. It is a right-handed helix with axial rise of 3.32 angstroms per base pair. It has a helical pitch of 30.97 angstroms and a base pair rotation of 38.58 degrees. cDNA has diameter of 19 angstroms, smaller than that of A and B form of DNA. The tilt of base is 7.8 degrees. Fifth type of DNA is ddNA. This form of DNA is formed when structure is devoid of guanine. It is one of the extremely rare forms of DNA. ddNA is right-handed helix and the number of base pairs per turn of spiral are only 8, with axial rise of 3.03 angstroms per base pair and tilt of 16.7 degrees from axis of helix. It has a helical pitch of 30.97 angstroms and a base pair rotation of 38.58 degrees. eDNA, also known as eccentric DNA or extended DNA, it has a long helical axis rise and base perpendicular to helical axes. It has a deep major groove and a shallow minor groove. eDNA is intermediate in crystallographic pathway from bDNA to aDNA. Seventh type of DNA is hDNA. hDNA is usually found in pyrimidine or polypurine segments that contain within themselves a mirror repeat. Now coming to ribonucleic acid. RNA, like DNA, is a long unbranched macromolecule consisting of nucleotides joined by 3-5- phosphodiester bonds. The sugar moiety in RNA to which the phosphate and the nitrogen bases are attached is ribose. Ribose contains a 2-hydroxyl group not present in deoxyribose. RNA contains the pyrimidine uracil in place of thymine, which is characteristic of DNA molecule. Uracil, like thymine, can form base pair with adenine by 2 hydrogen bonds. However, it lacks the methyl group present in thymine. The native RNA is single-stranded rather than a double-stranded helical structure characteristic of DNA. Since the RNA molecule is single-stranded and complementary to only one of the two strands of a gene, it need not have complementary base ratios. Like DNA, RNA also has different types. In all prokaryotic and eukaryotic organisms, 4 main types of RNAs are found: ribosomal, transfer, messenger, and heterogeneous nuclear RNAs. The 4 types of RNA molecules differ from each other by size, function, and general stability. Ribosomal RNA or insoluble RNA is the most stable form of RNA and is found in ribosomes. It has the highest molecular weight. Ribosomal RNA is most abundant of all types of RNAs and makes up about 80% of total RNA of the cell. The ribosomes in prokaryotes and those found in plastids and mitochondria of eukaryotes contain 3 different types of RNA: 23S RNA and 5S RNA in the larger, that is 50S subunit, and 16S RNA in the smaller, that is 30S subunit. The ribosomes of the eukaryotic cells also contain 3 kinds of RNA: 28S RNA and 5S RNA in the larger, that is 60S subunit, and 18S RNA in the smaller, that is 40S subunit. The 60S subunit contains a 5S rRNA, a 5.8S rRNA, formerly called 7S rRNA, and a 28S rRNA. Transfer RNA, tRNA or soluble RNA is second type of RNA. It is the smallest polymeric form of RNA. tRNA amounts to about 15% of total RNA of the cell. The tRNA molecules serve a number of functions, the most important of which is to act as specific carriers of activated amino acids to specific sites. Each tRNA is specific of an amino acid, that is, it can bind or accept only that particular amino acid. All tRNA molecules have a common design and consist of 3 folds, giving it a shape of a claw leaf with 4 arms. The longer tRNAs have a short 5th or extra arm. They contain from 7 to 15 unusual modified bases. Many of these unusual bases are methylated or dimethylated, derivatives of A, U, G and C. These include nucleotides of pseudouridine, various methylated adenines and guanines, methylated pyrimidines such as thymine, and 5-methylcytosine and others. The 5' end of tRNA is phosphorylated. The 5' terminal residue is usually guanylate. The base sequence at the 3' end of all tRNAs is CCA. All amino acids bind to this terminal adenosine. Another type of RNA is messenger RNA, mRNA or template RNA. Messenger RNA is most heterogeneous in size and stability among all the types of RNAs. It amounts to about 5% of total RNA of a cell. It is synthesized on surface of DNA template. Thus, it has base sequence complementary to DNA and carries genetic information or message, hence its nomenclature for the assembly of amino acids from DNA to ribosomes, the site of protein synthesis. The 5' end of mRNA is capped by a 7-methylguanosine triphosphate. The translation of mRNA into protein begins at the capped 5' end. The other end of most mRNA molecules, the 3' hydroxyl end, has attached a polymer of adenylate residues, 20 to 250 nucleotides in length. Fourth type of RNA is heterogeneous nuclear RNA or hnRNA. In mammalian cells, including those of human beings, a precursor RNA is first synthesized in the nucleoplasm by DNA-dependent RNA polymerase. This precursor RNA constitutes the 4th class of RNA molecules and is designated as heterogeneous nuclear RNA or hnRNA. hnRNA molecules appear to be processed to generate the mRNA molecules, which then enter the cytoplasm to serve as template for protein synthesis. RNA not only carries blueprint message from DNA to the factories that churn out proteins, but it catalyzes the reaction in the same way that protein enzymes do. The particular type of RNA acting as a biological catalyst has hence been called as riboenzyme or simply ribozyme. The ribozymes have the ability to act as a sort of molecular scissors. In certain eukaryotic cells, the mRNA does not enter the cytoplasm as a naked RNA strand, but often remains encased by certain proteins. These are termed as informosome. The proteins bound to informosomes provide stability to mRNA molecule and also protect it from degradation by the enzyme ribonuclease. So summing up, there are 2 classes of nucleic acids, DNA and RNA. Both DNA and RNA contain a combination of 4 different nucleotides. Each nucleotide is composed of a 5-carbon sugar, phosphate group and one of 4 nitrogen containing bases. The bases found in nucleotides are adenine, guanine, cytosine and thymine or uracil. The DNA structure that accounted for all the available data consists of 2 helical DNA chains wound around the same axis to form a right-handed double helix. The sugar-phosphate handrails form the 2 sides while the paired nitrogenous bases form the rungs. The space between the rungs is 0.34 nanometer, while the strand as a whole makes one complete turn of the helix for every 10 base pairs. A number of biochemical and biophysical techniques, particularly X-ray diffraction studies of crystalline DNA, have shown that double-stranded DNA can adopt a variety of conformations. These include local variations in the helical parameters of DNA, other helical species of DNA, and alternatives to the double helical structure. DNA duplex model proposed by Watson and Crick is right-handed spiral and is called BDNA or balanced DNA. In the model, the base pairs lie at nearly right angles to the axis of helix. B-form DNA is thought to represent the conformation of most DNA found in cells. X-ray analysis of DNA crystals at atomic resolution have revealed that DNA exhibits much more structural diversity than formerly envisaged. Such variants are A, C, D, E, H, and Z form DNA. Ribonucleic acid or RNA, like DNA, is a long unbranched macromolecule consisting of nucleotides joined by 3-5- phosphodiester bonds. The number of ribonucleotides in RNA ranges from as few as 75 to many thousands. In all prokaryotic and eukaryotic organisms, 4 main types of RNAs are found: ribosomal, transfer, messenger, and heterogeneous nuclear RNAs. Each of these polymeric forms serve as extremely important informational links between DNA, the master carrier of information, and proteins. RNA not only carries the message from DNA for protein synthesis but also catalyzes the reactions in the same way that enzymes do. These are known as ribozymes. In certain eukaryotic cells, the mRNA does not enter the cytoplasm as a naked RNA strand but often remains encased by certain proteins. These are termed as informosomes. The proteins bound to informosomes provide stability to mRNA molecule and also protect it from degradation by enzyme ribonuclease. So, students, with this we conclude our today's lecture. Hope you have enjoyed it and understood it well. Thank you.